Hello, everyone. I think we might make a start. We're running to a pretty strict schedule this evening. So I'd like to welcome you to the MHTP Student Information Night. And tonight you are joining into the Hudson Institute departmental information sessions. And the first one off the rank is the Centre for Cancer Research. And my name is Jason Kane. I'm a research group head in the centre, and I'm just going to give you a couple of minutes of quick information about how this webinar is going to run. Obviously, all of the attendees, you guys are muted at the moment. There will be opportunity for questions, but what we ask is that you actually type the questions into the chat function. And for the last 10 minutes from 5:20 to 5:30, we will go through all of these questions and address them individually. And hopefully we can answer anything that you have. The session is being recorded, so I expect that you'll be able to rewatch the session if you missed anything. And you're also welcome to attend multiple sessions. I hope you know that. So this is the CCR session. There's another 4 sessions to follow this, but they are on separate links. So you will need to log into a separate link to see those sessions. I have also just, if I hit enter now, I've just uploaded a link to the student booklet. So this is the Hudson information on student projects that are available for next year. So that should be in your chat function now. And there'll also be a link in a slide that's going to come a little bit later in this session. The link that I've just provided you has all the details of all the available projects and all the available, all the relevant labs. Specifically within CCR, you'll find a link for that, but also for all the other Hudson centres. And this will give you information about all the available projects, who the supervisors are, and how to contact them. And if you have very specific questions regarding any of those projects, I encourage you to contact the supervisor of that project directly via email, and then you can ask them directly. So now I am going to pass over to Ron Firestein, who is the head of the Cancer Centre, and he will give you a bit of information about what exactly we do in this centre. Over to you, Ron.
Thank you very much, Jason, and welcome to all the students joining us today. So as you can see in this slide, we are one of the 5 centers at the Hudson Institute of Medical Research, and the Cancer Center resides in the Translational Research Facility, which came about 5 years ago. And this is really a fruition of bringing together the Monash University, Monash Health, and the Hudson Institute in one singular building to facilitate both basic research as well as translational research. So one of the strengths and opportunities in our cancer center is really this co-location and ability to intersect with both clinicians as well as basic science researchers. And one aspect to that, of course, is having clinicians embedded within our center and working alongside them to really understand the clinical impact and translatability of our research. So I think that's one of the underlying themes that you'll hear about today, is really how to bring basic science into the clinic. So if we can go to the next slide. Great, thank you. So as I mentioned, within our center, we have a number of research groups, and you'll hear about some of those more specifically later on in this talk. But the general theme, I think, within our center is really bringing about translated cancer research through both innovative approaches as well as novel models of cancer. And these involve both mouse models that different groups are developing across different tumor types, as well as using technical approaches that are relatively new, like the CRISPR technology and high throughput drug screens, as well as genetic screens that enable us for the first time really to sift through thousands or tens of thousands of genes, identify new targets, and then validate them using clinically relevant models of cancer. And this gets into the overall theme of precision medicine. And as you'll see, a number of our students you'll be hearing from today have been involved in working through some of these new technologies to both identify and validate therapeutic targets dealing with really pressing problems in cancer, such as chemo resistance, as well as cancers where we currently don't have targeted therapies for them. So we can go on to the next slide. So I'd like to introduce Dan Goff, who's one of our research group heads focusing on stat cancer signaling, as well as more specifically lung cancer modeling, as well as pediatric cancer. So I'll leave it to you, Dan, to continue.
Thanks, Ron. So on this slide here, we're highlighting those group heads or mentors who are looking to recruit students for next year. And we've also just listed just a very brief description of their research focus. And you can see here that we do cover off quite a large variety of different tumour indications, but also favoured experimental approaches to really interrogate those tumours. And so just quickly, I'll run through these group heads and just in very brief terms give you an idea about the sorts of projects that are on offer. So Ron has interests in both paediatric cancer and also in colorectal cancer, and he uses a combination of precision medicine and also functional genomics to interrogate the fundamental mechanisms in those diseases. My lab, as Ron has already introduced to you, has interests in lung cancer, so primarily in small cell lung cancer, and also in paediatric cancers. But we also have a heavily biochemical side to the lab where we're looking at JAK-STAT signaling in cancer metabolism. Jason Kane has interests in both paediatric cancers and also in lung cancers. And the projects that he's offering for next year are really looking at epigenetic pathways and how they're regulated or can be targeted for the treatment of these types of tumours. Minnie's expertise is in RNA biology and also in colorectal cancers. And her projects for next year are really focusing on microRNA regulation and how that relates to colorectal cancers. George and Jim are our resident hematological malignancy experts. They have long-standing interests in NF-kappa B signaling and how that drives leukemias, as well as also in hemoglobinopathies, and both their projects reflect these long-standing interests. Andrew and Marie are our ovarian cancer experts, and their projects are really looking at the cancer stem cell phenomenon, cancer metastasis, the immune cancer microenvironment interactions, and how better to define new targeted therapies to treat ovarian cancers. And so if you see projects, or if you have particular interest in any of these areas, please look up the research projects in the book that the link has been provided to you already, and contact the supervisors directly to get more information about those projects. Jason, I'll get you to advance to the next slide. So on this slide here, I just want to tell you why you should come and work or do your training in the Centre for Cancer Research. And Ron's touched on some of these points earlier. One of the real key strengths, I think, here is the approach of translational research, where we take fundamental biology and try to understand the mechanisms that really go wrong to drive these tumours, but then to take that all the way through to the point where you're identifying new targets, how to target them, using preclinical mouse models to validate these targets, and then to partner with other groups to really see the impact on patients and their families' lives. And really, I guess the important point here is that we can't do research of this scope in isolation. So all of our research groups and group heads belong to much larger collaboratives, and so that's collaboratives nationally and internationally, and these teams are multidisciplinary. There's other researchers, there's clinicians, we have consumers, philanthropy, drug companies, you name it. really, it's that collection, that holistic sort of collection of skill sets that are required to be able to perform these types of research. And I guess the important point there is that your project, whilst it's discrete within the research team that you'll be joining, you'll be exposed to a much larger sort of array of types of research, and you can see how your discrete project can fit into a larger research program to really benefit patients. Now, Ron has gone through some of these technologies, but I just want to quickly touch on them again because I think that they're really critical to the way that you approach these sorts of questions. The things that we're trying to address here are obviously incredibly difficult, and to be able to do this, we need to really adopt new technologies and really embrace them as they come available. And so we have done that in a very large way in our department. We have very extensive access to CRISPR libraries, also to drug libraries, and high throughput capacity to be able to use both those screening technologies to really complement one another to identify new therapies and new targets. We also take a lot of advantage of mouse models and also patient-derived material, so both in terms of organoids and also in terms of sophisticated imaging modalities to look at the tumour development within mice. And so that can be as simple as sort of luminescent and fluorescent readouts, but also using really clinically applicable readouts like MRI, PET, CT, and I've got a PET CT scan of a lung cancer-bearing mouse behind me here on my screen. And so I would encourage you to consider joining CCR for the training environment that will provide you to really set you up for the next stage of your career. With that, I'm going to hand over to Shazia, who's an honours student in our department, who's going to give you a perspective on the honours program. Thank you.
Thank you, Dan. Hi, everyone. My name is Shazia, and as Dan said, I'm a current honours student in the Hudson Monash Pediatric Precision Medicine program that Ron mentioned before, also known as MHTP MP. So I'm supervised by Associate Professor Dr. Ron Feinstein and Dr. Paul Daniel. So currently there are many students undertaking really interesting projects on different types of pediatric brain tumors. In particular, I'm working on hybrid gliomas, which is a very aggressive type of brain tumor affecting adults and children. So my project focuses on comparing adult and pediatric hybrid gliomas in order to identify therapeutic targets. And this is done by utilizing really advanced techniques that Dan mentioned before, such as CRISPR, which is a well-known gene editing tool. This enables me to identify genetic vulnerabilities in adult and pediatric hybrid gliomas. And moreover, I conduct drug screens where I screen over 2,000 drugs from different drug libraries in order to identify drugs that cause a significant inhibitory effect in my adult or pediatric lines. So in general, honours is a very challenging year, as it's a very short period where you're assigned to conduct with lab experiments, write up theses, and prepare for presentations. And most importantly, you also have to concentrate on your coursework. I believe the secret to success here is time management. It's really important to allocate your time equally to all these divisions in order to become successful. So I have enjoyed every bit of my time so far at Hudson. It has been a very positive and a very friendly environment to work in. I'm actually very grateful for this opportunity, as I'm surrounded by a very intelligent and really creative group of scientists who are very supportive, and they really want you to do your best. There's absolutely not a day I actually regret pursuing my honours here. So I hope you're considering your honours here, because I think you learn a lot from the techniques that are done here. So I have had many undergrad students asking me if my supervisors are very easygoing, are they approachable? Well, in my point of view, definitely yes, specifically at the Center of Cancer Research. Whenever I have my doubts, my supervisors are always present physically or via email to guide me and advise me down the correct path. Also, apart from all the science, of course, there are many social events we organize. It's organized by the Hudson Student Society or the Center of Cancer Research. Recently, we had a trivia night, which is conducted by Jason Kane. However, Quinton will be who is speaking next. He'll be able to touch base on the social events as he has more experience about it. Also, I know many students, when they finish their undergrad, they're in doubt on what should I do next, or what honors project should I choose. Well, I suggest the first thing, if cancer research is something that you're interested in, make sure you read the booklets, which is there in the chat function, and just go through all the projects. And if there are many projects that interest you, make sure you email all the supervisors and meet them, and just visit and talk to the supervisors and ask them questions that that clears all your doubts. And whenever you make your final choice, make sure it's a project that you are willing to commit for a whole year. And is it an environment I would like to work on? Yeah, and I think those are the key aspects you need to consider when choosing your honors project. And I cannot reiterate more about Hudson. It has been, I'm so glad that I did my honors here. I mean, I have had an absolute great time, and thanks to my supervisors. So yeah, and I welcome you all join us, and I'm pretty sure you will have an excellent year. Now I'm gonna move on to Quinton, who is our PhD student who will be talking. He'll be able to give you more information about the PhD program, as well as the social aspects and more on.
Thanks, Shazia. Thanks. So hi, my name's Quinton, and I'm a third-year student, PhD student in Dr. Daniel Goff's laboratory at the Center of Cancer Research. And I'm here today to tell you a little bit about the PhD and master's experience here in the CCR. But first, I'll give you a little bit of an idea about the stuff that I'm currently working on. So my PhD project is about a type of lung cancer called small cell lung cancer, and it's the most lethal type of subtype of lung cancer. And the standard therapy for small cell lung cancer is chemotherapy, where the vast majority of patients actually respond really well. However, within just several months, the cancer always comes back and becomes resistant to the chemotherapy treatment. And when the disease comes back, it's almost pretty much always lethal. So my project investigates the reasons for the mechanisms of why this chemo resistance occurs. And one of the major approaches that I'm using is CRISPR gene editing technology. And this is where I can turn off specific genes within cancer cells to see how these genetic changes affects the response of cancer cells to chemotherapy treatment. And using the CRISPR technology we have here in the CCR, we can turn on or turn off tens, hundreds, or even thousands of genes simultaneously to identify genes that are able to modulate cancer cells' response to chemotherapy. So the end goal of my project is to identify genes that can drive chemo resistance in small cell lung cancer and use it as a drug target to overcome chemo resistance in a clinical setting. And here at the CCR, we have a suite of cancer cells and genetically engineered cancer mouse models to assess potential drug candidates or investigate genes of interest. So now I'll just talk a little bit about my student experience here. So it's been great so far. It's a really supportive environment with a lot of really intelligent researchers and students. Everyone is super friendly and are always willing to help or provide support, even between the different lab groups and centers. And we also have some close collaborations with Monash University and international collaborators as well. And there's opportunities to visit international labs and attend conferences. And we here at the CCR have a lot of social nights too and fun stuff going on, like morning teas, parties or events, even though all of that's been currently put on hold because of a ban on large gatherings. We had a really awesome Zoom trivia night that Shazia was touching on, which was hosted by Dr. Jason Kate, and that was really fun. Finally, I just want to mention that we in the Hudson also have a student society where they host events, dinners, and parties as well. And it's a good opportunity to get to know some of the other students across the different centers. And a highlight was the student retreat held in Phillip Island, and that takes place over 3 days, where you get to relax a little and just hang out with some of the other students and do fun stuff. So, I mean, overall, doing a master's or PhD in the Center of Cancer Research is great. You get to meet a lot of really cool people, make some new friends, and most importantly, we have some incredible science going on around here. So I urge you guys to check out the projects that we have on offer and just get in touch with any of the supervisors for the projects that you're interested in. Yeah.
Thank you very much, Quintin. Thank you, Shazia and Quintin, for sharing with us your student experiences. They were wonderful to hear from you. And this brings us to our last segment of this presentation, which is the Q&A session. I see there have been a couple of questions which have been already answered by Ron, but I'll read them out anyway. Does the CCR accept summer placements and internships? And of course, all centres do, and I think that's something you as a student would need to arrange with prospective supervisors. So that's a good thing to do over your summer break. The next question, wondering whether there are mid-year honours intakes, and of course there are. So thank you very much for answering those questions, Ron. And then finally, there's another question from, are these honours students able to be done with Bachelor of Science background? Because this person has a Bachelor of Science background in immunology and physiology. Of course, that seems to be the right background environment to be coming from. And of course, there's no problem in doing honours with that degree. Wondering if there are any other questions, please type them in as we discuss. I think for me, I can ask a few questions to Quintin. Quintin,
Yeah.
What stage of your PhD are you in at the moment?
My third year of my PhD,
Okay.
and we narrowed down some targets of chemo resistance that we're planning to drug really soon and take that into mouse models as well. So.
Okay, and have you discussed with your supervisor what the next stage of your career is?
Yeah, so I was thinking of doing a postdoc maybe, perhaps overseas or maybe in Australia as well, because I really love research and I just want to pursue research as a career.
Yeah, I can see the passion in you, Quintin. Good luck with that.
Thanks, Jim.
Any other questions coming through? There's a chat comment here. Someone can give me a hand. After doing masters from India in biotechnology, I want to enhance my application. Should I do honours or masters? Very good question. Research first. So I think that comment is, do I do honours or do I masters? I think there's a number of reasons why you would do honours first, or a number of reasons why you would need to do masters first. So, and that possibly is to do with giving you the right background and get you ready to do a PhD. Would any one of our panelists like to further add to that question? No? Okay. Dan, can I ask you a question? Do you think, what is your opinion about doing honours versus masters?
Sure. So for most of our domestic students, they would typically, the typical progression would be honours obviously immediately after the bachelor's qualification. And then that obviously gives them a snapshot of what life is like as a research scientist. And from there, they make the decision about whether to continue on with a graduate degree, be it a PhD or a master's, often is a PhD. And so that typically for the domestic students, the progression would be honours and then PhD. For many of our international students, they often perform a master's qualification before they come into the PhD program here. So it really depends on research experience, to be honest with you. So to get into the PhD program, you must have a dedicated research component to your studies. So either an honours year like we have here, or a master's that has a research component. It can be a coursework master's, but it has to have a research component, which has to be 25% of 1 year to be eligible for the PhD program.
Thank you very much, Dan, and that's very important. Getting you ready to do a PhD and having the right background is very important.
Jim, I don't know if you can see them, but there's a couple more questions that have come through.
Thank you. I just need to scroll down. Thank you very much. There's a question from Jane. Are there any student positions with a bioinformatics background or biotechnology? And the answer is yes. Anyone would like to add to that with regards to bioinformatics? Ron, you've got a bioinformatics.
Yeah, I'll just put a plug for the Pediatric Cancer Precision Medicine program. It's probably one of the more intensive bioinformatics program that we have in terms of building a large collection of genomic and functional dependencies for different types of pediatric cancers, and then integrating all of that. So that is a program that has an embedded bioinformatician, and also highly collaborative in working as part of large consortia and working groups to really integrate the data and identify computational approaches for sifting through those large data sets. So if anyone is interested in that, then happy to speak offline about the bioinformatics aspects.
Thank you very much, Ron. That is certainly, I know a lot of people are very interested in the area of bioinformatics and how that is being integrated in our clinical translational aspect of all of our research.
Jim, can you might be able to see some of the questions coming through. Do you mind if I just rip through a few quickly, just
Thank you,
in the last couple of minutes? So
Aye.
just on the Q&A, we've got, have COVID restrictions changed how things are run in the labs? Certainly it has to some degree, but at the moment, medical research is an essential service. And so at the Hudson, for most labs, and certainly mostly throughout CCR, all the students are currently working in the labs. They only go into the labs to do their wet lab bench work, and then all writing and reading, etc., is done from home. And all meetings at the moment are done via Zoom, similar to what we're doing now. But research is forging ahead, which is good. Is it possible to complete a PhD in 3 years? It is. You need to be very organised and work very hard, but it certainly is. You know, the aim is that most PhD students will complete in the 3 to 3.5 year mark, certainly by 4 at the latest, but certainly possible to do it in 3. Jim, did you have some that you wanted to answer, or you've already answered them? I'll keep going if you like. You can feel free to interrupt. For summer internships, do we just contact the supervisors? Yes, that is what I would suggest. If you, if there's a project or if there's a area of interest that you would like to work in, just get in contact with the supervisors via email. Are you able to do 3990 research projects with one of these groups? Again, contact the research groups that you would be interested in working in. To see if they will take students as part of the coursework. I do not have an immunology background. I came from a chemistry background, but still interested. Is this a disadvantage? Not at all. Any background to do with science is suitable for honours. We certainly have a whole lot of different programs within the centre, you know, ranging from bioinformatics all the way through to proteomics and some chemistry. So again, just touch base with the right people for the right projects. Question.
Jason.
Yeah, sorry, just 2 more quickly. I'll try and get through before we leave.
30 seconds.
Do we need to, what information do you need to provide the supervisors? Just touch base via email, and we're pretty friendly. We won't bite, and we'll try and direct you in the right directions. And maybe I'll just pass over to Jim, and I'll try and answer some of the others via text.
Thank you very much for reading those final questions, Jason. Well, that really brings us to the end of the Q&A session. And let me just remind you all to please, if you want to look into further research projects, the link is there on the page, and that's also accessible in the chat function. And you'll find the next session, which if you go to the next session, that's also available by clicking an active link in the chat function. So hopefully that's been put in there now. So thank you all for your attention, and we look forward to hearing from you. Bye, everyone.
Bye, thank you.